All right, so we started our new unit on it's still genetics, but this time we're gonna talk about like Punnett squares, pedigrees, a little bit more mutations. So today we're gonna focus on the just vocabulary, and then Punnett squares. Mendel's law of heredity. Gregor Mendel. So he was an Austrian monk who developed three principles of inheritance by conducting genetic experiences on pea plants. So on your test, if they ask, it's definitely pea plants. Um, so let's go to this vocabulary. Heredity. Heredity is inherit from parents to uh, to their offspring. Okay, so true breeding is offspring that is identical to the parents. Obviously, we're not true breeds because we don't look identical to our parents. We don't have identical stuff to our parents. Hybrid is what we are, offspring with parents with different traits. Because obviously, we have a mama and a daddy. Obviously, they're not the same. So we have cross pollination. So cross pollination transfers pollen. That's where the pollination comes from the anthers, okay, to the stigma, okay. So again, from the anthers to the stigma of a flower, this is going to increase genetic diversity because you see it right here. Oh, this little bee was hungry. Yum 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 yum. He was like, oh, there's another little plant over here. Yum 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 yum. But all inside of here, that's actually pollen. And so, of course, pollen is the way of uh genetics. It is the gametes for the plants. Well, self-pollination. Well, here is where the plant basically impregnates itself from right here. This is a type of lily. This decreases genetic diversity. Why? Well, because we look the same. You getting your own pollen and going into your own egg. So you getting your own sperm, putting into your own egg, making your own baby. Period. All right, P1 generation is the parent trait. F1 generation is the first filial generation, daughter or son. F2 generation is the second filial generation, daughter or son. So what I usually be like, grandma, mama and them, and you. Okay? But let's just look at here. Here go grandma and them. So grandma is short, grandpa tall. All right. Look at you. Your daddy came out tall, and then your mama was tall. They had some babies, and they all are tall because they got this trait from granddaddy. But that one kid came out short like their grandmama. Genes are segments of DNA that control a particular hereditary trait. So we can have a gene just for a particular trait, okay? Now, which occurs in pairs. The different forms of genes are called alleles. So just having one, this is a gene, be like, oh, this is blue eyes, and now we got both of them coming together. We got alleles. Yeah, alleles is going to be different forms of the gene. So you're going to have an eye gene, but the color of the eyes is your alleles. Okay? Dominant. Dominant, the allele masks the hidden trait. Dominant always wins. Recessive is what's masked. And what it's basically saying, dominant takes over. If somebody is dominant, they always got something to say. They try to control. They try to dominate. That's the whole point of dominant is it dominates. Recessive. So by the allele that is masked by this. So let's look at these dogs. They both look the same, right? But obviously they had 3 kids. This is a dominant trait. This is recessive because who came out with who had long hair? That's like your mama got brown eyes, your daddy got brown eyes, and then you come out with green eyes. That's recessive because Maybe your grandma got green eyes because your mama ain't got it and your daddy ain't got it, so it's not dominant. Now, this is what we have to know. Genotype is the genes. Genotype is the genetic makeup of an organism. Genetic. You do not see it. Okay? You don't see the genotype. Phenotype is off of physical appearance. So physical, okay, by how it looks. I'm going to look at you and say, oh, you got some brown eyes. Okay. I ain't going to look at you and say, oh, you got big B, little b. No, I'm not going to say that. Nobody's going to say that. Okay, if you say that, you need to go somewhere and sit down. Nobody talks like that. We be like, oh, you got some pretty green eyes. Is your genes big G, little g? No, we don't say stuff like that. So phenotype is what you see. Genotype is the stuff that we don't say. That's the genes. But here's the 3 principles from Mendel. Here's the very first one. So the principle of dominance and recessive. So obviously there's a dominant and a recessive. One factor in the pair of alleles may mask or hide another factor, preventing it from having an effect. Basically, again, here you see black corn, you don't see no yellow corn in it, and you have a yellow corn, you don't see no black corn in it. They had kids, they had one kid that came out black, but then their kids came out mixed. It doesn't matter, it's just one that hides it. So when it's either dominant or recessive, either I'm going to see a recessive trait or I'm going to see a dominant trait. We don't see both because it can't work as both. Here is the principle of segregation. So the alleles for a trait separate during the formation of egg and sperm. So as the egg and sperms meet, and we go through this, so of course you should know. Here's mom and daddy genes. Here is mom and daddy genes, and here's mom and daddy genes. But once they separate in the meiosis, we got the genes. Now the thing is, if it's a sperm cell and it connected to another cell, the egg cell, that maybe is the same, then it's going to be dominant. So this is a dominant gene, recessive, and recessive. That's pretty much how it breaks off. If your mama was big G, and your daddy was little g, most likely your cells is going to be big G, little g, and it's still going to walk in whatever was more dominant. How we do this is FOIL, first, outside, inside, last. But the principle independent assortment. So this is, again, when the genes from one trait will separate independently from genes from another. This is usually in dihybrid, which we actually don't learn about anymore, but this increases genetic diversity. This is more like for your hair color, your eye color, and it basically influences, they do not influence each other's inheritance. Now for me, there's one thing that I believe. When you see someone who has red hair, what color are their eyes usually? Green, right? Yeah. So if you usually have someone with blonde hair, you see them with blue eyes. Kind of adds up. So the thing is, the genes for hair color and eye color do not influence each other's inheritance. That's what it says. But I seem to differ. All right, so how are alleles written? So alleles dominant is big letters. Remember, they always try to win. They try to be the boss. So you always use the dominant letter first. Dominant alleles are always written first again. So big T, or big T, little t. Some people write it this way, but that's not how you're supposed to write it. Correctly is this. So if I ever say, hey, what's dominant? Write dominant. Okay. Recessives are always shown with little letters, okay? And the reason why this is dominant because the big letters first, just like you capitalize the sentence. And that causes it to be, as you see, dominant when we start off the sentence. Homozygous. Remember, homo means same, okay? So zygous, we have the same alleles. The gene pair of the alleles are identical. If you are a homo, you are the same. So that's identical. For example, big T, little t, little t. So if this is dominant, right? So that would be homozygous dominant. If I'm little t, then I am homozygous recessive. Because we're the same, so we're going to still be homo. Hetero means different. So obviously, that's the big letter, little letter. Because it's different. We don't like the same thing. So here's an example of crossing of two cats. So let's just say this is mom. Mom is big T, little t. So this is mom. She's red. Dad, he's big T, little t. He's blue. You see how I kept them together? Okay, cool. Now, what I'm going to do is take Dad's gene. Right? Then, I'm going to take Mama's gene. Because we're going to do big T times, big T is TT. Big T times little t. It's big T, little t, right? So right here, if I said how many were homozygous dominant? 1. That's that one, okay? If I say how many were heterozygous, remember that's different. Those then, it's 2. And how many was homo recessive? 1. So probabilities, y'all know probabilities. You all should know those by now. If you flip a coin, it's 50-50. So used to predict offspring with the crossing of two parents. It's 50-50. You're either going to have a boy or a girl, right? And if you have anything in between, that's a mutation because that's not normal. So then we have a test cross. Test cross is a cross of individual unknown genotype. So again, genotypes is what we don't see with the individual of a known genotype. And so in this case, homozygous recessive. Punnett Squares is developed by Reginald Punnett. This diagram is used to basically predict and compare genetic variations from a test cross. We're going to learn about monohybrid. I want to give you the definition of dihybrid, but we only teach monohybrid now. Here's your monohybrid. Cross between individuals that only have one pair of traits, so one pair. So we're only talking about, okay, this is the color of the flower. So big P times big P was this one. Came out purple. This one here came out purple because why? Purple obviously is what? Dominant. If that's what I'm seeing with big letters. And once you do the math again, you see it. And then here at the very end, this one came out white. That one, it came out recessive. Look at their two little letters, okay? And if I say, what's the ratio for the phenotype of purple to white? That's 3-to-1. So I'm going to do this with you. So look at this box. As you look at this box, you'll realize that the monohybrid is going from homo times homo. So this is homozygous dominant times homozygous recessive. I'm going to be honest, this will always be the percentages. So how many big Y, big Y? There was none, right? These are not bananas, they're peas. Big Y, little y. Well, homo times homo will always equal up that. No little y, little y. How many came out yellow? 4. How many came out green? So the ratio, this will always be the same. If I ever say, hey, what's the ratio from homozygous dominant to homozygous recessive, which is these. This ratio right now is 0 to 0, technically 4 to 0 ratio. All right, so homozygous times heterozygous. And again, this will always be the same ratios. So here is the homozygous, as you see, because it's the same. Is that homozygous dominant or recessive? Right, it's recessive. So in this right here is hetero because it's a big Y, little y. So here, how many big Y's? None. How many big Y, little y's? 2. How many little y's? 2. How many came out yellow? 2. How many came out green? 2. So this is a 1-to-1 ratio. Of course, homo times hetero. Then here we have hetero times hetero. So again, hetero is big letter, little letter. So how many big Y, 1. How many big Y, little y? 2. How many little y, little y? So this ratio is a 1-to-1 ratio. Now, yellow came out 3, and how many came out green is 1. Now, this is where the terms are coming to. If I say, what is the phenotype ratio? Phenotype is what we see, right? I'm going to see yellow or green, so that would be 3-to-1. But if I say, what's the genotype? Those, again, are letters. And then, just again, dihybrid just means it's 2 or more. And as you see, this has to do with a lot more. Like here, you see there's nothing up here in the color. For example, body color in the presence of hair. Just a practice question, the first phylogeneration is F1. The way an organism looks, phenotype, cross of an unknown genotype with an individual of a known genotype. Test cross. And the likelihood that a specific event will occur, probability. All right, have a good one.